Good morning, ladies and gentlemen. Welcome to session number 23, which will focus on theranostic concepts in cancer immunotherapy. I'm very happy that we managed to bring together a couple of leaders in the field, and I'm very much looking forward to their talks, which will certainly all be outstanding. I'll start off myself with a short introduction on the topic. So my name is Twan Lammers. My name is Twan Lammers. I work at the University Clinic of RWTH Aachen in Germany. And the title of my talk today will be Nanomedicine and Theranostics in the Era of Immunotherapy. So 2010 to 2020 clearly is the era of immunotherapy, I would say. In 2013, Science Magazine decided to make cancer immunotherapy the breakthrough of the year. 2018, the Nobel Prize for Physiology or Medicine was given to cancer immunotherapy, to Allison and Honjo for the discovery of cancer therapy by inhibition of negative immune regulation. And I think actually immunotherapy has been much older. So it's been dating back arguably to 1863 when Rudolf Virchow for the first time showed actually immune infiltrates in tumors. And then later on, Coley injected heat-inactivated bacteria into patients for immunotherapy. Multiple people followed up on cancer immunosurveillance, injection of tumor-infiltrating lymphocytes, and then later on also CAR T cells. So it's really a long history of cancer immunotherapy that we're dealing with. The landscape of cancer immunotherapy is very full. So this is an overview of ongoing clinical trials, and you really see huge numbers for different types of therapies, T-cell targeted immunomodulators, cell therapies, vaccines, viruses, and so on. And what you can also see if you look at the landscape is a tremendous, exponential increase. Comparing, for instance, the numbers from 2017 to 2020, what you then see is that in 2017, there were 2,000 agents with approximately 250 targets. And then in only 3 years, the number of agents more than doubled, and also the number of targets almost doubled. So it's really an exponentially developing field that is very much on vogue at the moment. The nanomedicine era started a little bit earlier. So I think the term nanomedicine was coined for the first time in 1991 in this book from Freitas et al. And then it started to become really popular around the year 2010, when Warren Chan and colleagues wrote this perspective article in the New England Journal of Medicine. And it was really hyped for a couple of years in the mid-2010s. So this is from the movie that you see streaming at the Clinon portal, in which nanomedicine was dubbed to be the next big thing in tech at the World Science Festival in 2013. If we look at the nanomedicine landscape, so obviously there are already quite a number of drug molecules out there that were nano, but that were not coined nanomedicine yet, particularly liposomes. But also here from the years 2000 and 2005 onwards, you really see a good increase in the number of abbreviated new drug applications and also investigational new drugs. In a very nice overview by Samir and Aaron in Bioengineering and Translational Medicine last year, they summarized all nanoparticles in the clinic, and they noted that there were, in 2016, 25 nanomedicine approved by the FDA or EMA, and 45 other nanoparticle technologies in clinical trials. And then in the 3 years that followed, 3 more nanoparticle drugs had been approved, 75 new trials started, and 50 new nanoparticles entered the clinic. So this is actually looking quite good. If we then turn to cancer nanomedicine, I think there's something like a controversy. So the cancer nanomedicine controversy, it reads like a big time title, but it's actually really an issue at the moment. Many people are discussing it from various different angles. I think one of the most critical papers was by Frank Zoka and Vendito, 2013, pointing that there are very many papers and only very few drugs in cancer nanomedicine. And it is in a way true. So if you look at Google and you just put cancer nanomedicine and you search for images, you see almost 6 million results popping up on cancer nanomedicine. There are more than 10 journals, tens of thousands of papers, many citations, and zillions of funding that have been spent on cancer nanomedicine. And actually, there are only 7 new drugs in the last 2 decades. And I think, I put only between quotation marks because I think it's not a bad number. It's actually quite a reasonable number, and many of those drugs are really adding a lot of benefit. But the question is whether this is in line with this significant spending and the very high interests that have been in cancer nanomedicine research. And I think in addition to this, so by definition, also antibody drug conjugates are falling under the definition nanomedicine. Dario Neri will be talking about this later. There are approximately 10 antibody drug conjugates currently used in the clinic and many more in clinical trials. And I think there are also a number of cancer nanomedicine that are really exploiting nanomedicine technology to in optimal form. Like for instance, Vixios, in which two drugs, daunorubicin and cytarabine, are encapsulated in a fixed ratio. So it's co-delivery of two different compounds in a nano formulation to really achieve unprecedented therapeutic efficacy in patients with leukemia. So the controversy continues. There was a 0.7% tumor accumulation paper by Warren Chan a couple of years ago, and then people were discussing whether nanomedicine has a delivery problem. And I think the delivery problem goes beyond just the percentage of tumor accumulation. It really goes in the direction of delivering what has been promised, right? So people raised a lot of very high expectations, and maybe we have not really lived up to those expectations. And very recently, Kenan Park wrote a paper in which he dubbed the cover story in Journal of Controlled Release the beginning of the end of the nanomedicine hype. And we had a very nice session at the annual meeting of the Controlled Release Society, the Stars Collide debate, in which big versus nano progress was being discussed. And there, Kenan actually voiced it quite clearly what he thinks about nanomedicine. So I'll share this with you.
Take a set nano and make it bigger.
So I do not think we need to make nanomedicine bigger, but we need to be looking at nanomedicine from a bigger picture perspective, if you ask me. And I think the time is now about right to start doing this and really look at the field and the progress more holistically, and try to figure out which areas we need to address to really promote progress in cancer nanomedicine and to make nanomedicine products more efficient in the clinic. I think there are a couple of things that we need to work on: patient stratification, drug selection or modular design. Combination therapy is very important, and definitely also immunomodulation. So I'll be briefly talking about these two areas, patient stratification and immunomodulation. Patient stratification in cancer nanomedicine, I think, will be crucial, especially if nanomedicine are meant to be delivering chemotherapeutic drugs in monotherapy or in combination therapy regimens. In order to make sure that they reach the clinic, that they progress from phase 1 to 2 to 3, we have to make sure that we select the right patients, because in all oncology trials, or the vast majority of oncology trials that are ongoing, patients are highly stratified. So tyrosine kinase inhibitors, antibodies are not given to 100% of patients, but only to 10 to 20% of patients, depending on receptor expression, certain genetic mutations. And I think in nanomedicine, we need to start considering that as well. So we need to use either imaging, or maybe even later on also tissue biomarkers to try to stratify patients, excluding somewhere between 30 and 70% of patients that are unlikely to accumulate nanomedicine in tumors, that are unlikely to respond, in order to make sure that clinical translation becomes more efficient. In cancer immunotherapy, this has already been done in the last couple of years. There are multiple ways on how you can stratify in cancer nanomedicine. In cancer immunotherapy. Most famous one is probably the Immunoscore by Jérôme Galland, who was supposed to be giving a talk in this session, but it might be that we need to skip his talk. There's also the cancer immunogram, which was proposed or established by people in the Netherlands, published in Science in 2016, and based on different features of patients or different features of tumors, we can predict reasonably well which patients are going to respond to which extent to different types of cancer immunotherapy. What has also been coming up in the last couple of years is a strong increase in the number of studies that have been combining nanomedicine with immunotherapy. There was a special issue in the Council of Chemical Research in 2019, which was entitled Nanomedicine and Beyond. And in that issue, my colleague Yang Xia and I, we looked at the combination of nanomedicine and immunotherapy. And what you really see is that this is very much en vogue at the moment. So in the 2000s, as discussed before, you see a very steep increase in the number of papers published on nanoparticles for drug delivery. In 2010, when the immunotherapy era started, you see a strong increase in the number of immunotherapy papers. Of course, you need to note that already before the 2000s, there was a good number of studies on immunotherapy ongoing. And then in the mid-2000s, you see this really steep increase in papers on the combination of nanoparticles and immunotherapy. So I think that in the next decade, we will be seeing a lot of studies, and actually already in this session, you'll be seeing quite a number of interesting studies on how you can use nanomedicine to improve the efficacy of cancer immunotherapy. If we think about principles of nano-immunotherapy, this is very simplistic and it's not complete, but there are multiple different things that you can do with nanomedicine in the context of immunotherapy. You can target chemotherapeutic drugs or toxins to cancer cells to induce immunogenic cell death, to make cancers better recognizable for immunotherapies in the immune system. You can target the tumor immune microenvironment to relieve immunosuppression, to, for instance, repolarize macrophages in the tumor microenvironment, making them more pro-immunotherapy. And obviously, we can also target the peripheral immune system in the spleen or in the lymph nodes to activate and engineer the body's own immune cells to start recognizing and killing cancer cells. There's been reasonably good progress in the combination of nanomedicine with immunotherapy. So you see this here in a review paper by Yang Shi, published early this year, where he summarized the number of clinical trials per nanomedicine product for a couple of selected products. So you see that with Abraxane, there are almost 100 trials ongoing in which the nanoparticle drug is combined with immunotherapy. Good number also for Doxil. mRNA vaccines are strongly coming up, particularly now also because of the current COVID situation. So that'll come back to the cancer situation likely in a couple of years, with a strong increase expected also there. And in the last couple of years, you also see this number of clinical trials that have been starting really steeply going up from 2015 onwards. And then finally, what I think what we're trying to achieve with nano-immunotherapy or with any type of priming before immunotherapy is that we start lifting up this survival curve. So immunotherapy has been a game changer because instead of just delaying the time till progression, as many chemotherapeutics actually do, depending obviously on the type of cancer, what immunotherapy can achieve is an increase in the number of complete cures. But still, the number is somewhere between 10 and 30, maybe 40%, depending on the type of cancer. And by using priming treatments, nano-immunopriming treatments, what we will hopefully be seeing in the next couple of years is that we will have drugs, nano-drugs, antibody-based drugs that can help to increase the number of long-term survivors upon treatment with immunotherapy. So with that, I've come to the end of my talk. In the interest of time, and I think also because this was a quite general introductory lecture, I don't think we will be taking any questions. I need to interact with the people at Clinic to ask whether Jérôme Galland is here. I believe he might not be. I can check whether there are any questions just to bridge time. There are no questions for the moment. If that is not the case, I propose that we move on to Jakob Kather. Is that okay? I'll stop sharing my screen and then Jakob can start sharing his screen. Jakob, please. So the next talk will then be by Jakob Kather from the University Clinic here in Aachen. So he'll be sitting a couple of 100 meters away from me at the moment. Jakob is a clinician scientist here at the Department of Medicine 3, studied medicine, also studied medical physics. And I think already early on in his training, he started working on neuronal networks and artificial intelligence. The title of his talk is Deep Learning-Based Histology Biomarkers in Solid Tumors. The talk will be 12 minutes, but since Jérôme is not there, Jakob, I would say we can go for 15 minutes, and then we'll take a couple of questions. Hopefully there will be a good number of questions and a good discussion. Please, the floor is yours.
Well, thank you very much. Thank you very much, Twan. Can you hear me?
Yes.
Okay, great. So I'm Jakob. As Twan said, I'm a clinician. In my day job, I specialize in patients with gastrointestinal cancer, but also here at University Hospital Aachen, I have a research group that's focused on deep learning based biomarkers in cancer. So this is not strictly nanomedicine, but I think there might be a good degree of overlap and maybe a good synergy in the future. So in the next few minutes, I'm going to talk about some fundamental stuff and also some recent data from our group. When I talk about deep learning-based image analysis, what I really mean is image classification. So that has been a standard problem in the field of computer vision. Imagine you have a bunch of different images that can be categorized into different classes. For example, cats and dogs, or whatever classes. And for humans, it's usually very, very easy to classify images into different categories. For computers, that used to be very, very hard for many decades, until deep learning came along. And now with deep learning, so more specifically deep convolutional neural networks, this task of classifying small images into different categories is very, very easy. And even nowadays, computers outperform humans in many of these very specific tasks. And the computers don't care if you which type of images are you're dealing with. So whether it's dogs and cats and dogs, or whether it's histological images. And so also with histological images, these methods work extremely well. And for us as clinicians, that's very, very interesting, because for every single patient with a solid tumor, we have a lot of histological image data that we can make use of. So how do we approach these problems usually? Well, that's a really straightforward workflow. You have a training step and you have an application step. Okay, so what we do is we collect images that have a particular label that we are interested in, a particular category. We use them to train a artificial neural network, and then subsequently apply this network to a bunch of images with unknown classes where we can predict the class. So in the context of cancer histology, as I said, we have a lot of images. Every single patient has cancer histology images available, and they usually look like this. So if you have a scanned whole slide image, that's really a huge image. That's like 50,000 by 50,000 pixels at least, and is just one single image is more than 1 gigabyte in size. So these images are much larger than other images outside of medicine, for example. So what we need to do in order to be able to process them is to cut them into smaller parts, and that's currently the standard in the field. We prepare a library of smaller image tiles that we can then pre-process, for example, rotate and color normalize, and then use for downstream analysis. If you have these image tiles, classifying these smaller images and derived from large histological images is not really hard. Okay, so there has even been a paper where they trained pigeons to classify these small image tiles into tumor or non-tumor. So that's really very easy, but it's also not very clinically useful, because in the clinic we are not interested in classifying small image tiles as tumor or non-tumor. What we really care about is the patients and the patient outcome and how we should treat the patients. So in the last few years, we've seen a number of publications in the deep learning histology field that have addressed clinically relevant questions on the patient level. So what we are interested in is, for individual patients, the most basic question is, does the patient have a malignant tumor or not? That's really not so difficult either, because pathologists can usually answer this question very, very fast and very reliably. In fact, pathologists are the gold standard for that in most tumor types. But of course, there have been a lot of papers in different tumor types where deep learning based image analysis could automate this and, for example, detect prostate cancer tissue in biopsies. However, in the clinic, other questions are much more interesting even. So, for example, take the tumor genotype. In the clinic, when we have a cancer patient, we want to know the genotype of the tumor, because in many tumor types, this guides our treatment decisions. So tumor genotype is a very important surrogate marker in the clinic. And indeed, there have been a number of studies in a number of tumor entities where deep learning-based image analysis could predict the tumor genotype just based on the histology images without additional molecular tests. And the idea behind that is that the tumor genotype, of course, shapes the tumor phenotype and changes the way the tumor looks. And just by looking at the image, you can basically infer the tumor genotype. Okay, but exceeding prediction of genotype from images, of course, in the clinic, we are even more interested in patient outcome and treatment response. So there have been a number of studies that predicted overall survival in cancer directly from histology images. And also now we've seen at least 2 studies that have predicted immunotherapy treatment response in melanoma and lung cancer directly from histology images. And I think this part will be the future. So hopefully we will see more studies that will use deep learning to mine this large amount of histology image data to directly predict treatment response. So now in the next few minutes, let me show you some examples of studies that came out in the last few years. And let me start again with colorectal cancer and prediction of overall survival. So in 2019, we published a study where we took 2 big patient cohorts of colorectal cancer. We used deep learning to classify the tissue into different classes and then put them together, put them into a survival model, and that could indeed, that model indeed was able to predict outcome pretty well in colorectal cancer. And our study was followed up by another team this year in a much bigger study where they took a lot of different cohorts from multiple countries, trained a neural network directly to predict outcome from histology, and that actually worked pretty well too. So that was an example of survival prediction in colorectal cancer with deep learning. In 2018, there was a very very interesting and fundamental paper published by a group from New York, where they, for the first time, showed that in lung cancer, they could use deep learning to infer genotype of lung cancer directly from histology images. So that paper inspired many other papers, including our own, which was published a year after, where we applied deep learning to colorectal cancer and showed that we can predict microsatellite instability, so a key genetic feature in colorectal cancer, directly from histology images. And how does that look like? So if we have a histological image and we cut it into smaller tiles and we then apply our prediction model, what we get is a heat map for the genotype, basically. And then to get a prediction for that particular patient, we can just average the heat map, or we can use more sophisticated methods to get from this heat map to a patient level prediction. What we see a lot in these heat maps is that the tumors have quite a lot of heterogeneity. So of course, that's no surprise. We all know that tumors are heterogeneous, but with most established molecular tests, you of course lose that spatial information, and you lose that information about heterogeneity, whereas image-based methods can visualize that heterogeneity and show that different parts of the tumor have different predicted genotypes in this case. So, what you have to do whenever you develop a new biomarker is, of course, large-scale validation. Otherwise, it's not going to be useful in the clinic. And for example, Jérôme Gallon, of course, did that with the Immunoscore in a big worldwide consortium. We also tried to we also built a multinational consortium to validate our deep learning-based biomarker and collected close to 10,000 patients with colorectal cancer with known microsatellite instability information, and then retrained a network on thousands of samples and externally validated that. And actually, the performance of this biomarker was pretty robust in this large study. So in the next steps, what we are going to do is to prospectively validate this biomarker. But of course, microsatellite instability is not the only genetic biomarker that we can predict based on routine histology images. So this year we've seen a handful of papers that have shown that actually in almost every type of solid tumor, there's a handful of clinically relevant genetic alterations that you can actually infer directly from histology images with deep learning. And one of these papers was our own, published a few months ago, where we showed that, for example, in colorectal cancer, there's a number of potentially clinically relevant driver genes whose genotype can be predicted from histology images with quite a good confidence. And not only mutational status of tumor driver genes, but also cytokine expression, presence of specific immune cell subpopulations, etc., was shown to be predictable based on histology images. So these images really carry a lot of information which deep learning can now extract, and which we are hoping to make use of as clinically relevant biomarkers in the future. So another study that was published actually alongside our own paper was from Cambridge in the from Kingston, Cambridge in the UK, and they also showed that that in a bunch of different tumors they can predict mutational status of TP53, BRAF, and many other clinically relevant genes directly from histology images, and also link that back to specific histological patterns that were not necessarily known before. So how many, if you want to do such a project, how many tissue slides do you actually need to train that project, to train your classifier? Well, in many small projects, we already start with 100 or maybe a couple of 100 slides, and that already gives us a good idea if that particular feature that we are interested in is predictable from histology images. But of course, the more images you have, the better. So in our study, we showed that if you increase your training set from 500 to 5,500 patients, your performance on the test set goes up. And also another group showed in a similar paper that if they train on 100, 1,000 and 10,000 images, their classification error went down, but there seems to be a plateau at maybe 10,000 histological images. How do we envision these methods to be implemented into clinical routine? Nowadays, at least in Germany, pathology workflows are not digital, so they really rely on glass slides and people going to a microscope and looking at these glass slides. But of course, in many countries, there are large-scale efforts underway to digitize routine pathology, and we hope that in a few years, of course, digital pathology will be the norm rather than the exception. And then what we can have is a workflow where a cancer patient gets a tissue sample taken, routine histology is performed, and at the same time, maybe genetic tests or other biomarkers are automatically triggered for that patient from the tissue, which can then subsequently be validated with molecular or genetic tests. And then all of that information can be put together in a tumor board, and a treatment recommendation can be given. So just as my last slide to give you an idea where the field might be moving, what I showed you is that we can really extract a lot of data from histology images alone. But of course, there have been initial papers that have shown that if you put together the whole slide images and other image features together with maybe genomic profiles, and you put that all together into a big matrix, and then you use machine learning to link that to clinical outcomes, that can even increase your improve your biomarkers additionally. So I'm very excited to see where this field will be going, and of course we are actively, our group is actively contributing to that. And what we really want to do is to build better biomarkers that are powered by deep learning that can answer clinically relevant questions. So that's it from my side. Thank you very much, and I'm open for your questions.
Thank you very much, Jakob. It was a really nice talk. Enjoyed it a lot. Let me see if there are any questions in the chat. Not yet. Willem, Dario, if you have questions, just speak up, raise your hand. I have one or two myself, actually. So what I was wondering, Jakob, so what feature do you think the computer is actually recognizing when you look at microsatellite instability?
Right, so with microsatellite instability, that's a very easy, that's a comparably easy case, because we know there are a number of image features associated with microsatellite instability, right? So that's tumor infiltrating, a high number of tumor infiltrating lymphocytes, mucinous tumors, poorly differentiated tumors, and stroma that is infiltrated by lymphocytes. So we know that there are these features that are associated with the genotype. However, in the past, it was not reliable enough to let pathologists quantify these features. And we think what the computer does is just giving us a tool to put together all these features and potentially others. So MSI is a good use case to demonstrate that this works, but really what I think is exciting is other studies such as that study or that study, where people could link, where we and others could link mutational status of, for example, TP53 to morphological features that were not known before. And what is a challenge right now is to decipher these features and to really look back into the trained deep neural network to learn which features exactly were considered by the network. But I think we can even learn about tumor biology from these approaches, because the network can show us and point us to regions in the tumors that are associated with specific genetic alterations.
Okay, Dario, you had a question?
Yes, if possible, yes, please, because in my very simple-minded view of how you do pharmacotherapy, because in the end, I thought this is what is useful. You have to choose between small molecules and, let's say, targeting otherwise of extracellular accessible targets. Now, if you go for intracellular targets, I think I see the logic. You need biopsies, you need to extract most of the information from your biopsies and maybe guide therapy. If you consider attacking the cells from the outside, be it with antibodies or with immunotherapy, what you really would like to do is not to take biopsies and actually use nuclear medicine as a surrogate. And I was wondering, you know, whether you see any day where nuclear medicine will replace the job of the pathologist, at least for what the accessible proteome is concerned.
Right, yeah, absolutely. So, of course, all of these procedures are invasive, and in a perfect world, we would have non-invasive biomarkers for most clinical decisions in oncology. And I would love to live in such a world. However, I mean, today, what we need to even make the diagnosis of a cancer in almost every single cancer type, in almost every single solid tumor, is a piece of the tissue. So colorectal cancer, prostate cancer, breast cancer, lung cancer, you have to have a piece of the tissue to even make the diagnosis. And our aim is to use that piece of tissue, which is available anyway, to extract more clinically useful information.
Perhaps looking at surface markers could still be important, and I know you do it, you know, HER2 new status, for example, because it will instruct, for example, antibody therapeutic options.
Absolutely, yeah.
Jakob, there are two questions from the online stream. So one is from Donald Bruce. How much can you depend on the classification if there are always going to be errors? So he's really asking about the errors in classification.
Right. Yeah, that's the big question. I mean, these can we really trust these things, or do we, or how good is that really? And the answer is we don't really know yet, because these methods are pretty new and the first papers were published 2 years ago. I think in some cases, such as MSI detection, what we are seeing is that the performance of these biomarkers is getting as good as the gold standard. So even the gold standard for MSI detection in colorectal cancer is not perfect, but has something like a 90-something% sensitivity and specificity, for example, in immunohistochemistry, and we are getting very close to that level. But even if we don't get perfect biomarkers, I think they will still be useful, because it would allow us to pre-screen patients. So our idea is that maybe you cannot test every single patient for rare genetic features such as NTRK fusions, but if we could use AI-based image analysis to bring down the number of candidates, maybe then we only have to test 10% of the patients for that rare genetic event, and that becomes much more feasible in our healthcare system. So we could use these methods to pre-screen patients before subsequent molecular tests, and that would be a very interesting clinical application, I think.
Mm-hmm. I have an in-between question myself. Do you foresee any issues regarding data sharing? Like ideally, we would want everybody to share their data on patients who have really good sets of data.
Right, we see a big issue with that. So most studies that were published work with the TCGA data, which was in the TCGA study, there was primarily focused on genetic data, and the histology images were provided in that study as a byproduct. And now a lot of studies are focusing on these images. what we see is that we need to build academic networks to collect a sufficient number of histological slides with associated clinical and genetic data to be able to advance these methods. And so that's actually the main challenge in the field, to build these networks and together to improve and validate these biomarkers.
Yeah. Good. There's one more question on the screen. Francisco Vasquez is asking whether you think that these technologies could replace conventional therapies, and likely also it means diagnostic protocols, or whether they would be in addition to, so in a way on top or in parallel.
Yeah, right. Again, a very, very interesting question. We don't really know the answer yet. I think once we have fully digitized pathology workflows, these methods, they don't cost anything if you just apply them to your images which are digitized anyway. So even if you could just use them as a pre-screening tool, that would be very, very interesting. But maybe in some circumscribed areas, we will also see these methods as definitive biomarkers. So I hope that the next few years will give us an answer on that.
Okay, Jakob, with that, I think we will be moving on. Thank you very much, particularly also for addressing really the keynote topic on digitalization and artificial intelligence. It was a really nice lecture. Thanks. We'll move on to Professor Dario Neri. We'll be starting his presentation in a minute. So Dario is a professor at the Department of Chemistry and Applied Biosciences at the ETH in Zurich. He's a pioneer in the development of antibodies and antibody-drug conjugates for the treatment of cancer and inflammatory disorders. He's received a number of awards for his work. Most recently, the World Antibody-Drug Conjugate Award last year. His talk is entitled Antibody Cytokine Fusions, and I think because Jérôme Galland ended up not showing up, I think, Dario, we can take 25 to 30 minutes for your talk and a couple of questions. Please, the floor is yours.
Twan, with great pleasure. Just a short question, please, because I have prepared actually a talk on antibody cytokine fusions as from the program. Mm-hmm. I also was not sure whether the audience would be more interested in antibody drug conjugates and small molecule drug conjugates. I have both presentations ready, so you just tell me what I should present, I click and I start. They are very different because in one case I focus only on cytokines as payload, and in the other case I discuss big versus small. You tell me what.
I think for this audience, big versus small would be more appropriate. It doesn't really fit to the session better, but I think the audience would be more appreciating.
Then I will do that immediately.
Okay, thank you.
Thanks a lot, Twan, and also Beat for the invitation and for the opportunity to present. And in this 20 minutes, 25 minutes, I would like to really contrast the use of antibodies or the use of small organic ligands as vehicles for the delivery of drugs. And this work was performed both at ETH Zurich, but also at Philogen, which is a company that I've co-founded 24 years ago. So basically, what we are discussing today is whether it's better to use an antibody, perhaps an antibody in IgG format for the delivery of a linker payload, or whether it's better to use a very small ligand that binds to cancer antigens and delivers the same linker payload. We have done work in both areas, and I think we can present some initial comparative evaluations. The problem to be solved is something that we all understand very well. Chemotherapy usually goes everywhere except to the tumor. And this is very well recognized in this picture. This is work that was performed at VUMC in the Netherlands. The authors labeled docetaxel, so a commonly used chemotherapeutic agent with carbon-11. They gave it to a patient with mesothelioma, and they basically watched where the drug goes. And the black spots tell you where the drug is located, and basically the drug goes everywhere except to the tumor. At early time points, you have activity in the blood pool, you have hepatobiliary clearance, which is already very active. Later, you have some uptake in the bone marrow. At later time points, you have excretion in the feces and in the urine, but at no time point a preferential accumulation in the tumor is visible. And this is actually a common feature of many different chemotherapeutic drugs and also of many different tumor types. And so many of us are still very fond of this idea of Paul Ehrlich, the vision of using magic bullets, Zauberkugel, in a way to use ligands for the active delivery of payloads, for example, drugs to the site of disease. Now, let's start with antibody-drug conjugates. Antibody-drug conjugates are one of the classes of armored antibodies that can be considered. So if you want to use an antibody as a delivery vehicle, and you can use a linker drug payload, but you may also consider cytokines that would have been the other topic for my presentation, or radionuclides or bispecific antibodies, or even CAR-Ts can be seen as armored versions of antibodies. So now we focus on ADCs. And the first thing we have to mention, and that was already mentioned by Twan in his introduction, is that actually now there are 9 ADC products on the market. So these are products that can be useful for the treatment of cancer in certain conditions. But there are also problems. If we go back to nuclear medicine and we look at how antibodies distribute in the body and reach the tumors, again, as a nuclear medicine study from the Netherlands, we see that the targeting process is very slow. Here we see a patient with head and neck cancer injected with a radiolabeled antibody in IgG format. At 1 hour, most of the activity is in the blood pool. At 1 day, most of the activity is in the blood pool. At 3 days, most of the activity is still in the blood pool, but you start to see the tumor. And why is that? Because antibodies in IgG formats are slow at extravasating. Their role is typically to stay in blood, not to go out and distribute in tissues. And sufficiently good tumor to organ ratios are typically achieved several days after intravenous administration. So this is a problem because it means that potentially we expose normal tissues to undesired concentrations of drug. Now, the problem is not only macroscopic, how much drug we deliver to the tumor, but it's also a microscopic problem. And this was well recognized by Genentech, one of the leaders in the ADC field, in a publication of 2007. You know that Genentech has developed an antibody-drug conjugate, Kadcyla, consisting of trastuzumab coupled to DM1, and they were looking at trastuzumab distribution within the tumor mass. And if you take a look at the tumor section from a mouse that was dosed with a very high dose, 10 mg per kg, the antibody is stained in green, the blood vessels are stained in red. You basically see that the antibody does not reach all tumor cells. The antibody only localizes to perivascular tumor cells, which are the ones that are reached first after extravasation. In other words, we have a macroscopic problem of targeting, but we have also a microscopic problem of not being able to reach every single tumor cell in vivo. Situation gets even more complicated when we think about the fate of the linker payload. And this is true both for antibody-drug conjugates and small molecule drug conjugates. We often postulate that the conjugates extravasate, they reach a target on the cancer cell surface, they internalize, and then the payload is released in some intracellular compartments and at some stage becomes available for its toxic activity. But there is a growing body of evidence that the linker payload may be cleaved extracellularly, and then the drug may enter also adjacent cells. This bystander effect is actually not a bad thing to have. And in the next slides, we will see how the linkers that are commonly used to produce ADCs and SMDCs are actually compatible with this mechanism. If you think most of the work in the ADC field features linkers between drug and antibody that either have a disulfide or an amide bond as a linker. And now, the moment that tumor cells start to die, they release glutathione or other reducing agents which can cleave this disulfide, or they release proteases which again can cleave peptide bonds. And so premature drug release can take place in the extracellular space, and this can be triggered by tumor cell death. And there is a beautiful review on this topic that was published in the British Journal of Cancer a few years ago. Now, we have shown in a number of models in the mouse with non-internalizing antibodies that, at least in the mouse, ADCs can work very well without internalization. This is one of the publications. Rémy Gebleu was the first author. He looked at tumor growth versus time. If you give saline, the tumor grows. If you give an irrelevant antibody called KSF, directed against HENEG lysozyme linked to the linker payload of Adcetris, so a cleavable linker and monomethyl auristatin E. Again, no therapeutic benefit. If you give the free drug at equimolar dose, again, no therapeutic benefit, whereas a targeted drug that goes to the tumor extracellular matrix and clearly a non-internalizing antibody specific to a splice variant of tenascin C could be potently active. So at least my opinion on ADC products is that they can offer an excellent selectivity in preclinical models of cancer, but they have problems. The cost of goods and the complexity of manufacturing is horrendous. We have seen that there are problems in the efficiency of targeting solid tumor masses. The distribution within the tumor is heterogeneous. And in many cases, we see good results in mice, but disappointing results in patients. So in other words, most of the time, we are not able to convert promising mouse data into equally promising therapeutic index in patients. And some people call this the holy grail of the field, really trying to understand how to bridge preclinical data and clinical results. Now let's consider small molecule drug conjugates. Small molecules have a nice feature that antibodies do not have. They can extravasate very rapidly. So in my experience, molecules smaller than 3,000 Daltons can extravasate in a matter of seconds. And this is nicely seen using intravital microscopy studies like this one, which is depicted, where in real time you can actually see how the small molecule floods the tissue. Obviously, if there is no binding event, the extravasation will be followed by the drug going back to blood. And that's why I said at the beginning, conventional chemotherapy, if you don't play a trick with ligands, will not go to the tumor, will not stay to the tumor. But the extravasation is a desired process. And if you link it to a suitable binding modality, then you can generate good small molecule drug conjugates. Now, there is a growing number of small ligands that have been validated for tumor targeting in vivo. We have done a lot of work in carbonic anhydrase 9, and for example, acetazolamide derivatives can target primary and metastatic renal cell carcinoma very efficiently in vivo. We have just published actually these findings in collaboration with a group in Vienna in the Journal of Nuclear Medicine. Uwe Haberkorn at the KFZ has pioneered ligands to fibroblast activation protein for the targeting of more than 30 different types of cancer. And you see these pictures, the selectivity is spectacular. And I will come back to this data in the final part of my presentation. PSMA is also a well-validated prostate cancer antigen, and the images are again wonderful, with some liability that you can see in the kidney and actually in salivary glands that may contribute to toxicity. But this type of targeting performance is rarely observed with antibodies. Now, both antibodies and small molecules can nowadays be discovered more easily thanks to encoded combinatorial libraries. My teacher, Sir Gregory Winter, and also George Smith won the Nobel Prize for Chemistry in 2018, also for their developments of antibody phage technology. And the technology is so powerful because you can build libraries with billions and billions of antibodies. The antibody fragments are displayed on the surface of a filamentous phage, and you go fishing these libraries for those binding specificities which bind to your target. And in my lab, we have libraries with about 100 billion antibody clones. Whenever we need an antibody, we just fish it out of the library, and the phage carries along the genetic information that can amplify those antibodies. And we have helped deliver similar solutions to the field of small organic molecules, where you use small ligands as protein binders and you use DNA tags as amplifiable identification barcodes. And I would like to show you this approach in more detail. This is a library that we published a couple of years ago with 35 million compounds. The molecules in the library can be visualized as dots in this cube, and their identity, their structure can be derived from this code 1, code 2, and code 3. This identified the building blocks that make up the molecule. A color scale tells you how frequent the individual molecules are in the library. And you can see that before selection, the molecules are uniformly distributed within the library, and after selection on 9 different antigens, you see different fingerprints. These fingerprints tell you basically which molecules you have fished out of the library, and then you resynthesize them without DNA and you use them for targeted delivery. Now, let's look at comparative evaluations of targeting tumors with antibodies or with small drugs. The first example I would like to present is based on carbonic anhydrase 9, and the second one is based on FAP. So carbonic anhydrase 9 is a validated tumor-associated antigen. It's a membrane-bound enzyme that can be drugged both by antibodies and by small molecules. There is excellent work that was started in the Netherlands again, but continued later at Memorial Sloan Kettering Cancer Center in New York. And basically, the investigators used an antibody called girentuximab, labeled with different types of radionuclides, and they acquired pictures like this, which are wonderful. The selectivity between renal cell cancer metastasis and normal tissues is very good, but this is typically observed only several days after injection for the reasons that were discussed before. Now, we have used many different carbonic anhydrase ligands, small ligands, to play the same game, but with small organic molecules. In this paper, you see, for example, that if you give a near-infrared fluorescent dye to mice with subcutaneous renal cell carcinoma, there is basically no targeting. After the initial distribution, you see clearance through the kidney, and at no moment in time you see a preferential accumulation in the tumor. If you couple the same payload to this carbonic anhydrase 9 binder, then you see, and this is an 8 nanomolar ligand, so it's a high affinity ligand. Already at 1 hour, you see a very clear localization in the tumor and with excellent selectivity, but the affinity is actually not enough to keep the drug on the tumor forever, but still the persistence is quite good, and I'll show you quantitation in a split second. And if you then use DNA-encoded chemical libraries to increase the affinity of your ligand and get a ultra high potent ligand, what we call acetazolamide plus, then not only we get good selectivity at 1 hour, but we get also long persistence on the tumor. Now, if we look at the same molecules, but this time with a radionuclide payload so that we can be quantitative, we can see that the acetazolamide ligand gives you about 18% injected dose per gram at 6 hours with excellent tumor-to-organ ratios. The higher affinity version gives you as much as 40% injected dose per gram in the tumor, but with some liability, especially in kidney and stomach. This can be reduced by increasing the dose of ligand, which is an interesting observation. Now, with these tools, Samuele Cazzamalli, at the time a PhD student in my lab and now group leader at Philogen, reported the first side-by-side comparison of an ADC and an SMDC. This is the structure of the antibody that he used. He mutated the cysteine residues in the hinge region to serines so that he would be left only with one reactive thiol at the C-terminus of the light chain, so he could get site-specific conjugation with this linker payload, which is the same linker payload as at Cetris. So you see valine citrulline with a self-immolating group, and then monomethyl auristatin E as payload. Or he used the same linker payload attached to this acetazolamide plus ligand that I've described in the previous slide. And what Samuele discovered is that, similar to the Genentech work, the antibody-drug conjugate could only target perivascular structures within the tumor mass. So the green staining tells you that you get a patchy uptake of the antibody in the tumor. If you use an antibody of irrelevant specificity, you do not see green, which means you don't see targeting. So the binding specificity was important in order to get some accumulation. But the situation with the small molecule drug conjugate was much better. Not only you had a greater uptake in the tumor in spite of a much shorter half-life in blood, but also the distribution within the tumor mass was homogeneous, and the experiments were performed at equimolar doses. Still working at equimolar doses, one could look at the therapeutic effect, and you can see that we gave 250 nanomoles per kg with the small molecule drug conjugate because the drug-ligand ratio was 1, whereas with the antibody we have a drug-antibody ratio of 2, and therefore we gave half the dose. You can see that the antibody can be potently active, but not terribly specific, in the sense that if you use an irrelevant antibody drug conjugate with no specificity in the mouse, this, at least in this setting, was also potently active, probably acting by slow release of a very potent payload. Whereas with the small molecule drug conjugate, I think we got a better discrimination. One of the surprises, at least to us, was that the ADC could be so potent in spite of the fact that the uptake was patchy, and also in terms of tolerability, I think the small molecule drug conjugate in red did very well. Now, Samuele has studied these small molecule drug conjugates at suboptimal doses in combination with an antibody cytokine fusion that we call L19IL2. It's a targeted version of interleukin-2. Again, at suboptimal doses, you can see that the single agents are not curative, whereas when you combine them, you get actually eradication of 100% of the mice in a model of renal cell carcinoma or in a colorectal cancer model. And when you look at what happens with the combination, you actually turn the tumors into a hematoma, and this is heavily infiltrated by natural killer cells. So probably the targeted cytotoxic creates the damage, and interleukin-2 boosts natural killer cells, which are still present in these immunocompromised mice. And in the last 2 or 3 slides of my presentation, I would like to show a second example that actually points in the direction of how good small ligands can be. The target is FAP. It has been dubbed as the next billion-dollar target for nuclear medicine. FAP is often found in the stroma of many different tumors. There were nuclear medicine studies published with anti-FAP antibodies, and I think the results were quite disappointing. And the work of Uwe Haberkorn actually really validated FAP as a target for small organic ligands. And these are pictures taken from his publication. And I've never seen actually pictures more beautiful than this. At early time points and with a long residence in the tumor, you get this type of selectivity. Look at colorectal cancer, look at breast cancer, ovarian, and other types of tumors, you really get the feeling that we may reach these targets in vivo with good selectivity. Now, our work has been to explore the chemistry of these conjugates, and we have found derivatives that we call OncoFAP, which to our knowledge are the highest affinity binders to FAP described so far. They also cross-react with mouse FAP. And when you go and perform quantitative biodistribution studies in tumor-bearing mice, the results are very promising. So first of all, if you look at the time course, look already after 10 minutes, you have more than 30% injected dose per gram in the tumor. The kidney values are already down at 10%, and the other organs are also low. And if you go to 1 hour, the tumor to organ ratios are just sensational. If you look at the dose dependence of targeting, you can see that basically you have a flat accumulation at 3 hours up to 1 micromole per kg. At 1 micromole per kg, you start saturating your target. And so this is my summary. It's a very subjective summary, but these are the lessons that we have learned over the years. I think both ADCs and SMDCs can be very active against mouse models of cancer. SMDCs may represent a convenient alternative to ADCs with lower cost of goods, better targeting performance, much faster development times. The discovery of novel small molecule drug conjugates is facilitated by advances in DNA-encoded chemical libraries. Both ADCs and SMDCs can be boosted using targeted cytokines, such as L19IL2. In our experience, both ADCs and SMDCs can work very well also without ligand internalization if suitable linker payloads are used, which are compatible with extracellular cleavage. And the efficient translation of these products from mouse to man remains a challenge and remains one of the most important problems that we have to address. Finally, I've mentioned the important role of Samuele Cazzamalli, who now runs the small molecule therapeutics group at Philogen. His collaborator, Jakob Millul, also importantly contributed to the project. Over the years, I've had a number of collaborators at ETH Zurich who have been important for ADC and SMDC development. Some of them have moved to Philogen, some others work at other institutions. And as a general thought, I would like to mention that small targeting agents are not only useful for the delivery of radionuclides or of drug conjugates. With chemistry that gets better, it is conceivable to generate bispecific molecules, which are basically functional equivalents of what you do with bispecific antibodies, but with much better tissue penetration and targeting performance. So I thank you for your consideration, and I'm glad to take questions if there are any.
Dario, thank you very much. Really nice lecture, very suitable for the purpose, I think. I'll start with 1 or 2 short questions, give people some time to put some questions in the chat. Do you consider these molecules nanomedicine? Right? It's probably semantics in the end, but they are, right? They have a carrier moiety, they have a drug molecule, the same with antibody drug conjugates. Would you classify them as nanomedicine? Some people do, some people don't.
Yes, I have no problems with, with the term because they are certainly in the nanometer scale. To me, I don't care so much about the names. I care about, I think there are two facts which are very important, I think. The first one is whether we get active or passive targeting. And we have discussed this in the past many times. And I know that passive targeting exists, the EPR effect exists, but in my experience, active targeting is more efficient. So I am sold to the concept of active targeting. If I want to achieve an efficient uptake, I typically need high affinity ligand. And the other concept, which at least to me is very important, is what is the size of the nanomedicine that you need? in my experience, if you want fast extravasation, you have to be below 3000 Daltons. And so this would reduce the size of nanomedicine to actually structures which are most of the time in the world of small organic molecules. But
can I follow up on that?
The jury is still out. I mean, the day that I see similar good targeting agents with larger objects, I change my mind.
Yes. Can I follow up? So how do you see the administration protocol? So should this be self-administration daily, like insulin? Or would it still be intravenously once weekly, maybe, because they clear so fast?
I think, you know, at least for cancer, administration will not be a problem, especially if a patient has porta-cut, you just want to inject. The real issue, but your question is an important one, and this depends really on the residence time on the tumor. So the small molecule drug conjugates go to the tumor in a matter of minutes. You have seen the data. What I think remains important is to really have very high affinity ligands so that they have a residence on the tumor of several days. And then there will be important research, and this is what the Journal of Controlled Release actually does, is to actually check, okay, provided you go to the tumor so rapidly, what is the optimal kinetic for the release of the payload? And I think for many of these questions, the rules have not been written. At least in my opinion, they have not been written, because you have to solve it one by one. You have to get good targeting agent, you need to get good payloads, and then you need to systematically explore how fast should the release be.
Mm-hmm. Okay, we have a question from Willem. Willem, maybe you can turn on the mic and the camera, just ask it yourself.
Okay. So it looks like I'm looking in the mirror, but okay. So excellent talk, Dario. I think that concept of targeting, that's, you know, targeting as a blunt statement, that's, you know, it's a little grandiose in a way, because there's, you can refer to tumor targeting. Maybe you would like to target, you know, cells that are in circulation. So do you have any experience in what type of context you prefer these drug conjugates for targeting, and maybe, you know, different types of contexts where you know, different platforms may be more suitable?
Yes, you know, I was trying to make, it's work in progress. So I don't claim to have definitive answers, and I've never been able so far to bring a product to the clinic. So we are at phase 3 with some of our products, so we're certainly not good enough. But I strongly believe that at least for solid tumors, most of the times we are not very efficient at getting there, not in humans. That's the only point I was trying to make. I mean, most of the times with conventional cytotoxics, even with protein-based therapeutics, we are not very efficient at building a tumor to organ ratio, which is good enough, which is the basis for everything we want to do. And so what I was proposing is that if you have small organic ligands with high affinity to good targets on solid tumors, I think this is as good as it gets in terms of targeting, in terms of biodistributions. And that's where the story begins, because then you have to build a therapeutic around it. And, you know, for example, there is, in my opinion, only one example in which a company has really systematically asked the question, is it better to use a radionuclide payload or a drug payload? So this was the work of Endocyte on PSMA. And in that particular competition, the radionuclide won, but, you know, it's N equal 1. Nobody tells us that if you use a different linker payload, you know, a different ligand, you get the same answer. So many words only to say that I don't think there are general solutions. I think the field of research, it's still in its infancy, but my gut feeling is already now with the tools that we have, we can probably improve over what was done before, either with drugs or with antibody drug conjugates. At least that's my opinion. Yeah,
and so to follow up a little bit on also what Twan alluded to. So do you think you see opportunity for these, I mean, I don't know if they're necessarily small drug, but these drug conjugates, for oral administration?
Honestly not. I think we are very far away from Lipinski rule of 5. We often use charges to keep these molecules out of the cells. So I don't see this as an area where you take it as oral drugs. I see this as a very clear opportunity for drugs that you inject intravenously. And the first challenge is to actually show that the SMDCs are better than ADCs. This has not yet been done. And so there's a long way to go.
All right, thanks.
Okay, Dario, you have a whole bunch of comments and questions in the chat. I'm going to pick just a couple. There are a lot of, nice compliments to your talk, saying as a former chemist, this was very easy to understand, essential points, great lecture. Matthias Bartz has a question that is related to what we were discussing. So what is the concentration dependence in the case of these molecules? So for radionuclides, obviously the dose is very low. You get very sharp signal to background. How do you see that for therapeutic drug concentrations?
This is a very good question. It's a very, very good point. It's not often presented. I showed in the FAP story, but we have published the same for carbonic anhydrase 9, that basically if you plot percent injected dose per gram versus dose in the tumor, at least in our hands, we get a flat line, which means your selectivity stays good until a certain threshold. And when you start stoichiometrically to saturate your target, then this drops. And so in the examples we have studied, there is no problem going down with the concentration, with the dose. But at some stage, once you have stoichiometrically saturated your antigen, then this is first year chemistry, you have saturated your antigen and your percent injected dose per gram goes down. So this has important implications. And that's why I think it's a very good question. Because with radionuclides, most of the time you can get a high specific activity and you don't care. You just use a dose that gives you a good imaging and you have a truly theranostic approach. Whereas for drug conjugates, it's not always so trivial that you can use a linker payload at the dose that gives you good targeting. And this is this trend towards more and more toxic drugs. I think it's a very important point. And so one has to be sure that the dose that you use is compatible with activity of the drug and no saturation of the target. And a lot of the work that I've seen in small molecule drug conjugates often goes, in my opinion, at concentrations where you have saturation of target and you have suboptimal biodistributions. So I think it's a technical but very important point.
Okay, one more technical question from Bruno Sarmento. He's asking what the yield of production of chemical conjugation is overall for antibodies or for the small molecule compounds. Is that good or reasonable?
This is nowadays with the technologies, this is not a problem. I mean, with antibodies, you can be quantitative. If you have antibodies with a single reactive thiol and you give a slight molar excess of malimido derivative, you get complete purity and really site-specific modification, chemically defined products. And obviously the same happens in the world of organic molecules. I mean, if you make your synthetic product, you get only the product that you want. And with small molecule drug conjugates, the cost of goods are negligibly low. So it's not really, for antibody drug conjugates, I would claim the process is complex. You have to set up GMP production of your antibody. You have to factor in maybe 100 mg of antibody per patient per dose. You have to have the GMP of the drug. You have to have the GMP of the conjugation. So that is something which, if the performance is there, you can justify all the expenses. But it's a big investment. It's a barrier to the market for whoever goes in the field. For small molecule drug conjugates, this is not the problem. The problem is making molecules which are good enough. And there are very few examples which have been studied and published so far.
Okay, and then the last one, I'm sorry for those attendees whose questions I cannot pick because there are too many. It's a question coming from Nathalie Mignet and also partially Stefan Halper and myself. So they're asking about the nature of the drug molecule. So Nathalie is asking whether polymer or lipid prodrugs would be more suitable. I was wondering, how much does it matter which drug you take? You want to have a drug that is actually also deposited once it's released. Right? So is that something you have thought about? I guess so, but.
Yeah, no, I think this is an important question, and this is also what people in the ADC field are asking. I mean, it's not that you have thousands of targets. You have tubulin poisons, okay? You have DNA damaging agents, okay? And looking into the future, you could dream of other types of payloads, you know, molecules that punch holes into cells or immune stimulators. But let's stick to these two categories. If you go with the tubulin poisons, I like them because you get a selectivity, you know, for example, all these molecules will clear through the kidney, but you don't have a lot of cell division in the kidneys. So again, going for tubulin poisons, you will build an extra biochemical selectivity between dividing cells in the tumor and less dividing cells, for example, in the excretion organs. What people have seen in the ADC field with DNA damaging agents is very potent drugs, but for example, with ADCs and DNA damaging agents, you see vascular damage. And so what does it mean? It means that probably you have continuous internalization of the ADC in the endothelium. You have this neonatal FC receptor recycling. And so almost in your molecule, you have a built-in toxicity if you are releasing your potent DNA damaging agent in the endothelium. So I think it's a very broad question to which nobody has the answer. We don't have that many tools. We use the ones that we have, and we try empirically to use the ones that work better. I don't have a general solution, but I know that we need ultra potent drugs. This I know for sure.
Okay, that's good. I'm going to ask one more. So Gert Storm just put a question which relates to what I was also wondering about, and then we'll really move on to Willem's lecture. Gert is asking whether you see hypersensitivity reactions like with other nanomedicine, and I was wondering whether you see kidney toxicity like with radionuclide peptides. What is the main toxicity?
So these are, again, very good questions. We have never seen hypersensitivity reactions with small molecule drug conjugates, and you don't expect it because these molecules don't present on MHC class 2. So they will not get peptide co-stimulation or T-cell co-stimulation. So I think, you know, like penicillin is the exception in hypersensitivity because you haptenate your protein. Otherwise, you know, ibuprofen, paracetamol, they are not immunogenic. Whereas the issue of off-target toxicity is a very important one. And Samuel and I have just submitted a paper to MCT where we have described a long-term follow-up, for example, of kidney histology, just to check that there is no delayed toxicity in the kidney. And again, with tubulin poisons, we don't see it. But this type of toxicity has been reported for radionuclide payloads. If you look at the PSMA targeting, the toxicity is what you expect from the picture. You have uptake in the salivary glands, you have uptake in the kidney, and with radionuclide, that's what you get. You get kidney toxicity, and you get actually very profound salivary gland toxicity. So again, I think theranostic approaches guide not only activity, but they inform us about safety.
Thank you, Dario. I think that's a great conclusion for this session on theranostic approaches. Thank you for a wonderful lecture. And we'll proceed to the last lecture of this session. Last but definitely not least, we're perfectly on time. So Willem Mulder, professor of radiology at Mount Sinai School of Medicine in New York, also professor of biomedical engineering in Eindhoven University in the Netherlands. He's made a number of really outstanding contributions to the field of nanoparticle imaging, nanobiologics, and nano-immunotherapy for the treatment of not only cancer, but also cardiovascular disorders. Willem, I would say 25 minutes plus a couple of minutes for questions. Please take the floor. Looking forward.
So the title in the conference program was therapeutic targeting of trained immunity. I added in cancer. So this is work that we're publishing tomorrow. It's still embargoed, but I'm very excited to share this work because you know, there's been 4 years in the making. I've a disclosure. I'm a scientific founder and board member of Trained Therapeutics Discovery. Okay, so this is a story not about targeting a tumor to get anti-tumor responses, but to use targeting of the hematopoietic system to rebalance the immune system and prime it better, or let's say prepare it better for anti-tumor responses. And so this story starts with a traditional paradigm in which, in the host defense system, in which, you know, we're all very much aware of right now during this COVID pandemic, that, you know, the first line of defense is innate immunity, which is fast, it's effective, it's non-specific, but it lacks immunological memory. And when an infection is ongoing, then ultimately the adaptive immune responses will help out. That takes a while, so 10 to 14 days. It's specific and it builds immunological memory. So this is a classical view of the host immune response. But this paradigm has been challenged because when you zoom in on the myeloid cell compartment, so the cells that orchestrate the innate immune response, what you just saw on the left, if you zoom in on these cells, work from Mihai Netea has actually uncovered that there is some kind of very primitive memory located in our innate immune system. And so the first response, or the innate immune response, which has always been considered to be non-specific, it's been, you know, I think this was postulated by Mihai Netea about 10 years ago, that there may be some primitive immune memory located within the innate immune system, but this is not specific, and it's regulated very differently through metabolic and epigenetic changes. Okay, so I won't walk you through this slide exactly, but it kind of, because I can't really see the screen, my own screen that well. No, I'll try to do it this way. But what you see here is that in let's say the activation or the sensing of specific pathogens, all kinds of molecular processes in myeloid cells are changed, are rewired. So it started as metabolic rewiring of immune cells, and these metabolic changes underlie epigenetic changes, and that allows an increased responsiveness or a better responsiveness of these cells to subsequent infections. And so this has been termed trained immunity. So it's essentially a very primitive innate immune memory. It's non-specific, but it's real. Let me go to the next slide. So this is particularly relevant during the current COVID pandemic, because what is becoming increasingly clear now is that young people actually experience increased resistance to the SARS-CoV-2 infection because of their, let's say, more naive innate immune responses. In older people, so people of our age and older, the innate immune responses have been muted by, you know, lifelong of exposure to pathogens. In young people, the innate immune responses are very effective, and they basically, just through innate immune responses, they allow the clearance of the virus. So one strategy that Mihai Netea has proposed is to artificially induce improved innate immune responses by BCG vaccination through the induction of trained immunity. So this is postulated. There's at least 15 trials going on worldwide with the BCG vaccine, and, you know, to see if that can protect against COVID-19. But a paper that Mihai Netea published with colleagues from Greece about 2 months ago actually shows that the strategy works in elderly patients. So what they've done is they vaccinated elderly patients with BCG and followed these patients for 3 years. And what they've observed is that in elderly patients, they observed a 80% reduction in respiratory tract infections. And so that's pretty spectacular, since a lot of these respiratory tract infections are caused by coronaviruses. You know, it makes sense to, you know, to look at this as a mitigation strategy in the current pandemic. Okay, but, you know, BCG is a living microbe. It's administered intradermally. It causes a little infection in the skin. It takes about 4 to 6 weeks to resolve. But that means that this is not a strategy that can be applied intravenously. And so as a vaccination strategy, this may be very effective. But if you would like to exploit trained immunity as a therapeutic strategy, you have to come up with more clever approaches. So let me walk you through how trained immunity is preserved systemically. So trained immunity is the metabolic and epigenetic rewiring of myeloid cells. That's what you see on the left. So a myeloid cells gets exposed to a pathogen associated molecular pattern. So that can be a real infection. It can be of bacterial origin, it can be fungus, but it can also be done, through something like the BCG vaccine. So that causes metabolic and epigenetic changes. And then when these cells, let's say, go back to normal, you let these cells rest. There's specific histone marks. So there's a specific rewiring of the epigenome that makes these cells more responsive to re-stimulation, to a secondary insult, which can be unrelated to the initial insult, and that's what makes trained immunity non-specific. So this is what's going on a cellular level. But if you take into consideration that, for example, monocytes have a lifespan of maybe a couple of days, that this cannot explain why these trained immunity effects are observed, for a couple of years in patients, let's say 2 to 3 years. That's because these properties they become part of the hematopoietic progenitors in the bone marrow, and these progenitor cells, let's say the parents and grandparents of immune cells, they adopt this trained immunity phenotype, and so they start producing myeloid cells that have that are epigenetically rewired. So there's something to the bone marrow. So can we design BCG mimicking therapeutics that nicely engage the bone marrow and then directly engage myeloid progenitor cells in the bone marrow? And that's what we do, and that's what we've been working on. We've been working on a platform that we call nanobiologics. The reason we call these nanobiologics is because these are nanomaterials that are constructed from our body's own building blocks. So these nanobiologics as is are completely benign. We can engineer them, we can make them in different shapes and sizes. And because of the integration of apolipoprotein A1, they also have very nice myeloid cell engagement through the interaction with ABCA1, ABCG1 that's shown here. So again, these materials are composed of natural building blocks, and so they by themselves are completely benign. And as I mentioned, we've created libraries, different sizes and different morphologies. And so now we wanted to adopt this platform as a strategy for cancer treatment, for cancer immunotherapy. And so in this case, we don't want to vaccinate to prevent an infection or maybe even to prevent malignancies, but here we want to have a therapeutic strategy based on the induction of trained immunity using our nanobiologics through the engagement of the bone marrow and bone marrow progenitor cells. So this is work that's been executed by actually 2 physicians that are working on their PhD theses, Bram Priem and Mandy van Leent. They primarily, Bram Priem primarily works in Amsterdam, also in New York, and Mandy van Leent works in New York in the group. And so essentially what we've done is we've taken our nanobiologic platform. The BCG vaccine is a live microbe, and so we identified in the BCG vaccine what triggers trained immunity, what induces trained immunity, and that's what we basically integrated in our nanobiologics. So you end up essentially with an artificial or the nano-sized microbe that also has the ability to induce trained immunity. Then we've done a lot of work on screening and optimizing. In the interest of time, I left out these data because I would really like to focus on the biology. It's going to be a little bit unusual. Normally, I would give more of like conceptual talks. This is really going into the details of how this exactly works, because this is pretty different, a new paradigm that really nicely complements what's already out there in immunotherapy. So let's focus again on that bone marrow. So the trained immunity can be induced systemically in the bone marrow by rewiring hematopoietic stem cells and multipotent progenitor cells. These cells are the parents and grandparents of immune cells, of lymphocytes, and of myeloid cells. And in case of cancer, what we observe is that the cancer protects itself against lymphocytes, so against the adaptive immune system, through using, in a disadvantaged way, the innate immune system, these myeloid cells. And so the tumors get an immunosuppressive microenvironment, which is composed of innate immune cells, of myeloid cells, that actually work against the host, but with the tumor and against the adaptive immune system. And that's one of the problems obviously in cancer and oncology. Okay, so we have then developed these nanobiologics. We identified a lead candidate. What you see in that nanobiologics, in this case, are discoidal. But what we decorated them with is peptidoglycan derivatives that we identified to be responsible for the induction of trained immunity of the BCG vaccine. Okay, now on top of these slides, you'll see the data or the concept animated, and then on the bottom are the actual data. And so what you see here is these nanobiologics. We've done cryo-EM and we measured their size. And then we can intravenously administer these nanobiologics in mice. We radio label these materials so we can quantitatively establish blood half-life. That's relatively short, but that's because of the engagement with the immune system, and so that's by design. And because we radio label these materials, we can also perform PET imaging. And as you can observe, there's a lot of accumulation in the bone marrow. Obviously, the liver, that always lights up, but relatively to the liver, we see very good bone marrow engagement. Okay, so what we've also done is intravital microscopy. This is in tumor-bearing mice, B16F10 mouse melanoma model. And what you see with intravital microscopy, this is microscopy on live mice. We've done it on the skull of mice. As you can see, the accumulation of nanobiologics in the bone marrow. We also see the accumulation in the tumor, but we're not looking for tumor targeting. And so this is just something, that you get as an additive, but that's not what we're focusing on. It's not that important also for the therapeutic responses. Okay, so what happens next? So these materials accumulate in the bone marrow, and then we see engagement with multipotent progenitors, but also with hematopoietic stem cells that we've teased out using flow cytometry protocols and flow sorting. And then the next thing that happens is that through NOT2 activation, that's a pathogen recognizing receptor, we see this metabolic and epigenetic rewiring. The metabolic rewiring we probe with in vivo FDG PET imaging. So we see increased glucose consumption in the bone marrow as a result of the induction of trained immunity using our nanobiologic immunotherapeutics. This epigenetic rewiring also, is associated or maybe even underlies epigenetic changes that we have probed using ATAC sequencing. And so we flow sorted hematopoietic stem cells and multipotent progenitor cells, and then we performed ATAC sequencing of these cells, and we see that there is, significant changes in the epigenome of these cells, particularly if you focus on processes that are related to innate immune responses and trained immunity, like interferon gamma production, TNF production, interleukin-6 production, and also the production bias towards myeloid cell and myelopoiesis. So what does this do to these cells? Well, we see increased proliferation of the hematopoietic stem cells or particular subsets of multipotent progenitor cells. And so that's what you see here. There's also through flow cytometry protocols, you see massive increase in proliferation. And that obviously results in higher numbers of these hematopoietic stem cells and the multipotent progenitors. We also teased out what subsets, but I left that out, for reasons of clarity. But there's specific subsets of multipotent progenitors that proliferate, that have an increased proliferation rate, and that kind of drives the production bias of the myeloid cell compartment. So then we've isolated the bone marrow from these mice, And then we've exposed these, we put these cells in culture and then we expose them to LPS restimulation. So the concept of trained immunity is that you have one trigger and then you have a subsequent trigger, and that relates, there you would observe increased responsiveness. And indeed, cells from the bone marrow of mice that were treated with our nanobiologic immunotherapy are more responsive to LPS restimulation in vitro. And so these cells that are being produced by the bone marrow now have this trained phenotype. We've also visualized it using CD11b PET imaging. CD11b is a marker for myeloid cells, and then you can see on a systems level with PET imaging, you see increased numbers of myeloid cells deposited in hematopoietic organs. So this is actually a very powerful tool and very nicely complements what you can do with flow cytometry and other techniques. And I think it's pretty unique. It was also the topic of today's session, like how can you integrate imaging for immunotherapeutic purposes? I think this is a beautiful example of how at least we've pursued that. Okay. And then when you focus on the tumor microenvironment, you see a rebalancing of that tumor microenvironment, where you basically go from a very immunosuppressive tumor microenvironment to a rebalanced, more anti-tumor type of tumor microenvironment. This is a flow cytometry protocol with 23 different channels, and when we use these meta clusters, and so you can use algorithms, so it's completely non-biased, but you can beautifully see these changes in the tumor microenvironment, and that really ultimately leads to dramatic reduction in these tumor-associated macrophages, and that cause immunosuppressive nature of tumors, and that ultimately then leads to tumor remission, as you can see here. So we've used the B16F10 mouse melanoma model. It's notoriously known to be very poor responder to immunotherapy, particularly its checkpoint inhibition refractory model. So it has very low responsiveness to checkpoint inhibition. And using our approach, we can nicely see, depending on the regimen, so the times we dose and the concentration at which we dose, that you can see beautiful dose response in terms of tumor growth inhibition. And here you see the actual mice from these groups, and so large tumors and very, very tiny tumors in the treated animals. I want to emphasize here, these mice had established tumors of significant size, and then we start treatment. And so it's not a vaccination strategy, it's really therapeutic strategy. Okay, so one of the questions is, okay, how are you so sure that these effects are perpetuated by the bone marrow? And the obvious experiment is to do bone marrow transplantation. So you take one mouse, you treat these mice, or you don't treat these mice, but then you collect the bone marrow cells of these mice. You irradiate different mice, and then you do the bone marrow transplantation, let these animals recover, and they do tumor inoculation, and you can see that effect, the anti-tumor properties of the immune system are preserved as is shown here in decreased tumor growth rate. And so this really shows, we can't exclude there's other mechanisms and there's maybe direct effects on the tumors, but the focus on the bone marrow here is, yes, it seems to be very, very significant. Okay, so currently, obviously, immunotherapy has, and there was also Twan's introduction. Immunotherapy is a revolution in at least in cancer treatment. Has really changed the way we think of how we can best help cancer patients, but also other patients that are suffering from diseases where there's a dysregulated immune response. And so there's two very well-known checkpoint inhibition drugs. One is focused on CTLA-4. That's one checkpoint, and the other checkpoint is PD-1. You also have PD-1 ligand. But so what we've done is basically investigated if the induction of trained immunity in combination with these types of immunotherapeutic strategies could work really well. As I mentioned a couple of slides back, this mouse model is checkpoint inhibition refractory. And so that means it does not respond well or does not respond to checkpoint inhibition, this specific mouse model. So here you see on the right, you see these black dotted lines that are basically control mice that are treated with PBS. And then if you treat these animals, and that's the yellow line, if you treat these animals with a combination of anti-PD-1 and anti-CTLA-4, there's no effect on tumor growth whatsoever. However, if you first induce trained immunity using our nanobiologic immunotherapy, then we can actually get complete tumor remission. And so the lowest line that you see actually demonstrates, it looks like there's still some growth, but there's actually groups of 10 mice, and 5 out of 10 mice, there was complete tumor remission. And so this very powerful strategy, because you remove the immunosuppressive barrier, and then if you then activate T cells with checkpoint inhibition, you get very potent anti-tumor effects. All right, so one of the missions of my lab is also, to move away from mice after we've teased out the therapeutic effects, and that we know that concept has a lot of potential. And we also do work in non-human primates, and then we integrate imaging. We use a PET combined with MR to look at biodistribution, and we also do some preliminary safety work. And so there's an overview of a summary of the data. So these are the same nanobiologic immunotherapeutics that we've used in mice, and then we've applied this in non-human primates. And I think everyone can agree with me that the bone marrow and the spine lights up like a Christmas tree. And so these properties, these bone marrow engaging properties of these nanobiologics are preserved across species. And we see this in mice. We also see this in rabbits and pigs, and here in a non-human primate, it's a primate that is getting very close to human situation. And you also see this relatively high bone marrow engagement as we quantified, analyzing these PET imaging data. What you see on the bottom right, we looked at some markers for kidney and liver toxicity. And you see that it's all within tolerable levels, and so this therapeutic can also be safely applied. Has very good tox profile. Okay, so to summarize, I don't know how much time I have, but to summarize, so our approach is not to target a tumor or target a disease, but actually to engage the hematopoietic system and to regulate the immune response using these nanobiologics. These are intravenously applied, and then they accumulate in the bone marrow. What I've shown now is the effects of the induction of trained immunity, which is particularly good in cancer, but it can also be applied in infection or sepsis. But you could also do the exact opposite. You could also try to manage or control the immune response by doing the exact opposite. And that can find application in cardiovascular disease. And we have many examples, many papers where we show this approach to be very effective in atherosclerotic disease animal models. Then as a little bit of an encore, because I was told that I could talk a little bit longer. I want to show you that we have done the exact opposite strategy of what we do in cancer and transplantation. It's just a couple of slides. So in transplantation, you could think of it's the other side of the immunological coin of cancer. In transplantation, there's an immune response, and that causes rejection of the transplanted organ. In cancer, you basically have an immune system that doesn't take care, is too tolerant towards that cancer. You would like to inverse these two situations. So we published a paper on this concept in Immunity in 2018. We made some advances, so we kind of worked on the second generation versions of our nanobiologics. Again, we found some specific materials that have very good bone marrow engagement. And then we use a transplantation mouse model where we transplant a heart from a BALB/C mouse into a C57 Black 6 mouse in the abdomen. So these mice have two hearts now, and the heart in the abdomen is connected to the abdominal aorta. So blood flows through it, but it doesn't have function. And then through palpation, you can feel rejection. And typically in this model, that happens at day 8. And so what we've done here is we applied these, anti-inflammatory nanobiologics before rejection, so at the day of transplantation, at day 2 and day 5, and then you can see that it really promotes allograft acceptance in this specific mouse model. So remember, this does not require immunosuppression. It just requires 3 injections at the day of transplantation, and then you're within 1 week after transplantation to additional injections, and that we see allograft prolongation pretty spectacularly. Yeah, that specific nanobiologic therapy we've also applied in non-human primates. Also found, that these properties, the in vivo properties of these materials were preserved, and that they also had a pretty good safety profile. Yeah, and so that kind of concludes my talk. So you can go both ways. The focus was on cancer, and then I'd like to end with acknowledging all the people involved in the work, folks at Mount Sinai, and my lab at Mount Sinai, obviously also in Amsterdam, collaborators, and at the Eindhoven University of Technology.
Thank you. Awesome talk, beautiful presentation. I have a first question to get things going. So I would expect that you could use BCG as a very simple adjuvant actually in many of those ongoing trials with checkpoint blockers. Why aren't people doing that?
Yeah, because they are.
Oh, they are. Okay, good.
Yeah.
So,
should I continue sharing my screen, or how do we do this?
No, it will be camera now. So
okay. No, I mean, there's also, trials of beta glucan also induces trained immunity. Beta glucan is, kind of like activates the fungal pathway of trained immunity. Yeah, so there's many initiatives. But in the current pandemic, I think it actually will make sense to just start BCG vaccinating people. It's actually well, I don't know. So, to some level, it's bizarre that they're very cautious about the BCG trials, and then we need to wait for the actual results of the trials. Well, this is a vaccine that has a it's 100 years old with a perfect safety record, and it also, if you think about it conceptually, the vaccines that are currently being developed for COVID-19, they are adaptive immune system vaccines, the traditional vaccines. But the elderly are maybe helped much better, if you boost their innate immune responses. So I think it will be great strategy in many different contexts.
Just to mention it is used for bladder cancer, right? I guess you know that.
And that's probably also through the induction of trained immunity. And the reason that, because it stays confined, right? So you can have it intravesicular, and then you don't get sepsis. But obviously you can't intravenously administer BCG. Yeah. But that's why we built these technologies.
Question from my colleague, Yang She. He's asking a related question, asking whether TLR agonists and things like that, do you expect they would also do something with innate immunity, like trained innate immunity?
So this is a new field and it's rapidly growing. And so there's a bunch of pathways that have now been linked to trained immunity. And it's rapidly growing. So I'm not aware of actual studies that show this, but I would not be surprised. So the pathways that are currently well identified is through DECTIN-1, and that's a pathogen recognizing receptor on NOC2. It's also shown that OX-LDL induces trained immunity, and there's lots of recent papers that show that there's probably a lot more PAMPs and DAMPs that induce trained immunity. So I'm not aware of any studies that conclusively show this, but I think it's a little bit in the line of expectation that.
Okay, good. Dario, please.
Thanks a lot. I was curious to better understand at the molecular level how you see the activity of BCG vaccination against COVID. Is it mainly related to the possibility to elevate the cytokine levels, and then indirectly they would activate both innate and adaptive immunity, or how would that work?
Okay, so we don't know yet, obviously, but what it seems to be, okay, so you basically control cytokine responses at the level of bone marrow progenitors, and that's trained immunity. And so, the BCG vaccine is actually, it's intradermally. It's a little infection that lasts for 4 to 6 weeks, and so that you have a lot of time to get, exposure to this specific pathogen. And that results, and there's literature on it, that results in epigenetic changes in the progenitor compartment. And so then the production of immune cells by that progenitor compartment, that are essentially immune cells that have a different cytokine response. And they have a much stronger cytokine response. And so that should result in 2 things, enhanced resistance of myeloid cells against pathogens, but also, potentially also better priming of the adaptive immune response.
I think I can follow this. I thought that in the end, the executioner, or at least the key mediators, would be increase the cytokine levels, and indirectly then they would modulate the activity of leukocytes. At least that's what I got also from your slide and from the rest of your presentation. I wanted to make sure I had understood it well. Sorry.
No problem.
Okay, so Willem, also here a lot of compliments for your talk. I need to select 3 short questions, short answers, please, and then we need to get closing. First one comes from Wafa Al-Jamal. She's asking, how long does the effect of trained immunity last in vivo?
Yeah, in a mouse, not that long. And so that's maybe 2 months, in that range, 1 to 2 months. In human subjects, up to 2-3 years. And so you see, for example, that BCG trial that was published in Cell. They followed these patients for 3 years, and they seem to have benefits up until 3 years.
Okay. Jan Niklas, my colleague, is asking whether you or why you see so strong uptake in the spine and not so much, for instance, in the hip or other large.
Yeah, that's a good one. And so in young animals, you see it in the femur and the tibia. And in older animals, you more see it in the spine. And apparently it's related, while we're aging, that's where, your bone marrow is more active. Yeah.
Okay, interesting.
And these non-human primates are not the youngest.
Yeah. One related question from Donald Bruce, who's an ethicist. He's asking about the animal models. What extra information do you get from using non-human primates over, for instance, pigs or other animals? For us, the most important reason is that, with any of these immunotherapies, before you go to patients, non-human primate work is typically done for regulatory reasons and tox work. There's not a major difference, but pigs are really big. So there's also a practical reason. The pigs are about like 75, 80 kilograms, small pig. And these non-human primates, they're about 10 kilograms. So that also helps in the logistics of what we do. But, obviously, these primates most closely resemble humans. Okay. Question from Jay Prakash, Twente, saying, excellent talk. Do you know if nanobiologics accumulate in macrophages or generally in myeloid cells? And did you check the status of macrophages in cancerous lesions, more M1-ish or M2-ish?
Yeah, so a macrophage is obviously used to be a monocyte. So we also see it in tumor-associated macrophages, but we don't really care for that too much. It kind of polarizes these cells in the same direction. That makes them a little bit more inflammatory, but these are not long-lasting effects. And so we've profiled. I recommend that if people are very interested in this, tomorrow this paper will be published. I recommend that people read that paper. But we looked at all myeloid cells, except not too much looked at neutrophils. And we also think that neutrophils are very important in these effects. And so we mostly looked at monocytes, macrophages.
Okay, with that, I think we need to get closing. Willem, thank you very much. Just like to mention that Jérôme Gallon excused himself. So apparently he changed secretary and it wasn't in his agenda. It's a pity that he could not join. But still, we filled the time very well. I think we had a very extensive and good discussion. Thank you, Willem, Jakob, Dario, for the great talks. And before closing, let me please refer you to the talk at 10:45, which will be given by Laura Kelly. We'll talk about her journey as a pancreatic cancer patient. This will really be an outstanding lecture and a journey of which I'm sure you will never forget it. So please make sure to rejoin us for session 24 at 10:45 in 15 minutes. Thank you.